Hello, everyone. I'm Kevin Kunzman, Managing Editor of HCP Live, here for another video conference interview as part of our ongoing ARVO 2020 coverage, with the data being split across an entire month, differing dates for exactly what sort of discussions we'd be having on our ARVO coverage. We're joined today by Dr. Stephen Russell, who is the principal investigator for a 12-month phase 1b/2 trial assessing patients with LCA 10 treated with Sepofarsen, showing significant improvements in visual acuity. And before we get into more of the endpoints and the findings from that trial, Dr. Russell, how are you doing?
Pretty good. I think we're all hunkered down.
That's great to hear. That's very reassuring at this time. So thank you so much for joining us as part of our ongoing ARVO coverage. So let's talk a little bit, if we can, Doc, before we get into the trial itself. Can you tell us a little bit about the investigative therapy at hand here?
Sure. So this is a rather novel treatment for the eye, using a antisense oligonucleotide to treat a genetic disorder, specifically the CEP290 mutation-associated Leber's congenital amaurosis, so a very narrow category of disease. And we can talk a little bit about the mechanism if you wish, but this particular drug is a small molecule administered as an intravitreal injection, which is familiar to most eye professionals, clinicians specifically. And so it doesn't require any additional technology or technique, such as the other forms of gene manipulation or gene therapy, like subretinal injections.
Right. And what exactly were the endpoints of this assessment as a phase 1b/2?
So as a phase 1, we were looking, essentially every endpoint was an exploratory endpoint. We had both pediatric patients and adults, and so there was a significant limitation on what we were able to measure. These patients, as most clinicians that treat them know, have nystagmus and very poor vision. This is a category of Leber's congenital amaurosis that has extraordinarily poor vision from very early on. And so things that we thought might not be very good endpoints turned out to be pretty good endpoints, and things that we thought would be good endpoints turned out to be not very good. It was kind of a discovery process as we went along. Interestingly, visual acuity turned out to be very important, as well as light sensitivity. These patients, of course, have a congenital form of retinitis pigmentosa, the LCA, so we were looking essentially at what their minimum detectable light intensity is as an endpoint, the full field light sensitivity, or FST.
Right. Right. And as I understand it, there are no currently approved therapies for LCA. I mean, what exactly have we seen, I guess, from previous assessment into treating it before that emphasized the need for these endpoints?
So it's without understanding the sort of the structure of how the diseases are referred to. So there is a treatment for an LCA, which is the RPE65 associated LCA, but there are no treatments for any other forms of LCA or specific treatments for retinitis pigmentosa. So this is a new gene. This is the CEP290 gene. And so, there's no currently approved FDA therapy for this. So this would be a novel indication for treatment within LCA. Would be potentially the second form that could be treatable.
And what's the patient population look like? Because I know you said adults and children, and it being a genetic disease. What's the reach of it, and what are they generally going through in their progression toward blindness?
Patients with LCA typically have very early onset poor vision. This is one of the poorer prognosis and more accelerated courses of patients that have LCA. So oftentimes they are legally blind by the time you can measure them, and progress to, not uncommon for them to progress to no light perception by the time they're of legal age. So it's a very rapid deterioration that occurs in this patient population.
Wow. Okay. And so what were the trial findings? What were the end results?
We had statistically significant improvement in visual acuity. The visual acuity improvements on average were about 4 to 5 lines of visual improvement, which is very significant. We also had a very significant improvement in the full field light sensitivity, so ability to be able to perceive in the dark, or in a dimmer environment, I should say, probably not dark, but dimmer environment. And then the third interesting finding was that we weren't able to reach statistical significance using a mobility test, which is the test that had been used, a test similar to what was used with the RPE65 gene therapy study. But there may be reasons why that may be, and that has to do with the differences in the course, and also the differences in the disease, and of course the differences in the therapy itself.